Welcome to Genetics 8, Basis of Selection. And we're going to cover several different ways that animals can be selected. We can select very young animals just based on their pedigree. We can select animals based on their own individual performance. Or we can get a bunch of progeny and rank bulls or boars or stallions based on how their progeny perform. The animals are going to be quite old before you get those progeny records. We can calculate estimated breeding values based on performance of all the relatives. And getting a little more modern now, we can do DNA tests on individuals and genomically enhance their EPVs. So that's what this little video is going to be all about. We'll start off first and looking at pedigree selection. So pedigree selection. Selection is based on performance or estimated transmitting ability of the ancestors. So we have a couple pedigrees here. We have two bull calves, Joey. His sire has a genetic evaluation of plus 1,000 pounds for milk. The dam has a genetic evaluation of plus 500 pounds of milk. So his pedigree index is the average of the PTAs of the parents for plus 750. Now Ben is another bull calf. His sire is better, plus 2,000 pounds of milk. Dam, same as the other one, plus 500. But we take the average of those two values, the average of the parents' genetics. So his index is plus 1250. So if we need to select a bull calf, obviously we're going to select Ben based upon his higher pedigree index. Now there are advantages and disadvantages to each of these methods. Advantage of pedigree selection is that we can do it early in animal's life. In fact, embryos are bought and sold based upon that pedigree index. It's also relatively cheap. We've got the pedigrees there. All we have to do is average the breeding value of the parents. Now there are some disadvantages. You have relatively low accuracy because you do not know what the sample of genes each parent may have contributed. And for example, there can be a lot of genetic variation between full sibs. And full sibs basically share 25% of their genetics on average. So it's a pedigree index, a way to rank animals. You can do it early, you can do it cheaply, but you have to realize that the accuracy may not be all that high. Our next basis for selection is based on an animal's own performance. And sometimes it's known as mass selection, because we can have a big group of animals and we select based upon their performance. So here we have a bunch of pigs and we can weigh them at the beginning of a performance period. We can maybe do some ultrasound, get some carcass measurements, and we have determined that these 4 pigs are the best. So basically relatively simple and straightforward, just selecting on an individual's own performance. So advantages and disadvantages for this method, you don't even need pedigrees. We're just basing on the individual's own performance. Now, pedigrees are really nice to have, and we can even maybe take into account the pedigrees on these animals to maybe avoid inbreeding later on, but relatively simple. Need a minimum number of records. And if it's a cheap measurement to take, we can measure a large number of animals. Okay, disadvantage, well, if the heritability is moderate or high, you can make quite a bit of genetic progress, but if the heritability is low, such as reproductive traits or disease-resistant traits, with a low heritability, it's really hard to make very much progress from mass selection. And traits have to be measured on each individual. You have to wait till the individuals are old enough to have performance data. Next we have progeny testing, most often utilized in dairy cattle and beef cattle genetic selection programs. Progeny testing became popular back in the 1960s, so it's been around for a while. So we have this bull, and we may have selected this bull based on his pedigree. We're going to distribute semen out to a lot of different herds. So each of these little pods here represents offspring in different herds. And then we compare those offspring to offspring from other sires that have sired animals in those herds. And as I say, progeny testing has been very popular on an organized level from the 1960s. Advantages, disadvantages of progeny testing. And biggest advantage, it can be extremely accurate way to rank individuals. In fact, you can achieve 99% accuracy even on lowly heritable traits if you get enough offspring. Now, on lowly heritable traits, you need hundreds of offspring, but you can rank those animals very accurately and make genetic progress. It can be used for sex-limited traits, and there's a lot of sex-limited traits, especially traits that are only expressed by the females. So important sex-limited traits would be milk production in dairy cattle and dairy goats. Other sex-limited traits would be litter size in swine. Only the females have litters to count. So basically we can progeny test those males, see how their female offspring produce, and then select our males for further propagation based upon how the females produce for those sex-limited traits. So big advantages here. But there are some disadvantages as well. It's time consuming. We have to distribute out the semen and then wait till their offspring are born. If it's a case such as milk production, the offspring are born, then they don't start producing milk until 2 years of age. We're not going to get a lactation record complete until those animals are 3 years of age. So for dairy bulls, it takes about 5 years time. You got to house the bull all that time. You got to collect the data, and we used to say always that it cost about $60,000 to progeny test one dairy bull. And at the end of the progeny test, then we could rank those bulls, and usually about 1 out of 6 bulls would be shown to be good enough to be put back into active service, while the other 5 bulls would be just sold to slaughter at a minimum return there. So it's kind of expensive, but it does work. The other thing, because of the expense, you can only, a limited number of individuals can be progeny tested. And for example, in the dairy industry, we have 9 million dairy cows. And so each of those cows would have a calf each year, half males and half females. So you're going to expect over 4 million bull calves being born. but we can only really afford out of those 4 million bull calves to progeny test about 1,500. So you have to choose those bulls to be progeny tested very carefully. Usually we're going to select those bulls based on their pedigree index. So those bulls initially selected on pedigree and then progeny tested. So it's kind of a two-stage selection process if we're going to do progeny testing. Okay, the next basis for selection is then the calculating estimated breeding values from information on all relatives. So we have this individual here who we want to calculate a breeding value. So we've got performance of his sire, his mother. We may have progeny. We may have some siblings. This bull here may have had hundreds of offspring. We may have some maternal relatives as well. So we're going to take all that information into account, put that data into a computer. The computer is going to weight all that information. Obviously, closely related relatives are going to put more, have more weighting than distantly related relatives. In livestock production, we generally report these genetic evaluations as transmitting ability. We're most concerned about how much these selected individuals are going to transmit to their offspring. An individual transmits half the genes, so the estimated transmitting ability is 1/2 the genetic value or breeding value. And to make things a little confusing, we use different terminology for different species. For poultry, some breeds of horses and dogs, we consider it estimated transmitting abilities. PTAs for dairy cattle and goats, EPDs for beef and swine. And for sheep, sheep actually rank their, the sheep industry ranks sheep based upon the estimated breeding value or full genetic value. So they don't use transmitting abilities for sheep, but rather than genetic values for ranking the animals. And the transmitting ability, of course, just 1/2 the breeding value. Whether you multiply a number by 1/2 or multiply it by 2, your animals are still going to rank the same. It's just the numbers are going to be bigger or smaller. So again, ETA's estimated transmitting ability used in the poultry industry. Most breeds of horses don't have organized computerized genetic evaluations, but a few do. They generally refer to them as ETAs. In dairy cattle and dairy goats, known as PTAs or predicted transmitting ability, and you might ask, why does every species call their genetic evaluation numbers by a different name? And it's really because these genetic evaluations for the different species were developed independently. You'll get a committee of people together and they may say, let's call our genetic evaluations something different. In beef, cattle, and pigs, they're known as EPDs, expected progeny differences. And again, it's the half of the parents' chromosomes that are passed on to the offspring. Now with sheep, they're known as EBVs, not transmitting abilities, but the animal's full genetic values. And one reason that sheep are different than other species is that the data from the performance of sheep is sent to Australia, where the genetic evaluations are completed. So in a lot of foreign or countries other than the U.S., they rank their animals on EBVs, estimated breeding values or estimated genetic values, rather than estimated transmitting abilities. Well, what are the advantages and disadvantages of calculating estimated breeding values, EPDs, PTAs, etc., for these animals? Well, it's most accurate if you can utilize all the information. So we're utilizing information from all the relatives rather than just the individual himself or a few progenies. Now the disadvantages need sophisticated computer program. You need excellent pedigree records. So you really do need a good record keeping system. You need to know the sire and the dam and paternal grandsire, paternal granddam, etc. You need a lot of good information. And because it is a lot of information, it really helps if all the breeders cooperate to provide good information. Finally, development that's been occurring since about 2009. 2009 genomics kind of came to the forefront. Genomics refers to using genetic markers or single nucleotide polymorphisms to help estimate an animal's genetic value. So we're actually looking at the biology. What are the genes and pieces of chromosome that's inherited from father to offspring, mother to offspring? What is the value of that piece of DNA? So with genomic testing, you'll hear the term SNPs, single nucleotide polymorphism. A single DNA test can identify differences in thousands of single bases spread across the genome. And just to illustrate real briefly, here we've got a chromosome, and at this loci here we've got a base, and there's 3 billion bases spread across the whole chromosome. So this nucleotide here may be a A. This nucleotide here would be a B, and we don't do all 3 billion, but we do thousands, so this particular section may be inherited intact from parent to offspring. And we might know from our records that if an A is inherited, it's going to be worth 10 pounds more milk than a T, if a T is inherited. And of course, this information is spread across the whole genome, and each of these SNPs has a very small effect for these quantitative traits, but when you're evaluating 50,000, you can really spread out the genetic evaluations on your animals and also increase the accuracy of evaluation at the same time. Just for you, a little demonstration here. This is genotypic data for Elevation. Elevation was a bull that was born in the 1960s. They were able to do a genomic evaluation or a DNA test on him from semen that has been frozen from that time. So 50 years after the semen was collected, we got a DNA test on Elevation, long after he was no longer living, of course. Well, this is on chromosome 1, and this is the data that goes into the computer. So we know at this particular loci, he is, for the bases, he's got an A and a T. So any place you see a 1, it means that this bull is heterozygous at that particular location. If it's a 0, he's homozygous for one particular base, 2, he's homozygous for the other. But this also just points out on the extreme amount of genetic variation there is out there to work with. SNP effects for milk production, and we evaluate a lot of different traits. Milk production, of course, is an important one, but here we have the chromosomes across the bottom, chromosome 1, chromosome 2, chromosome 3, chromosome 29 out here. And we'll just draw a line right here. And so below the line gets a little noisy, I guess, but above the line will show you genes that have major effects on milk production. Obviously, there's a gene on chromosome 14 with a major effect. But there are genes on every chromosome that does have an effect for milk production. So advantages of this whole genomics thing, and obviously there's some advantages. We've been able to make faster genetic progress in a lot of our species. So it adds to the accuracy of the other methods. So, first you're generally going to estimate the EBVs on the animals. We're going to do that first. And we take the genomics test. So especially in the beef cattle industry, they call it genomically enhanced EPDs. So the EPDs are more accurate because of the DNA test. Can be used to estimate genetic values on traits not usually measured, and we're doing a lot of work there. We still need to collect phenotypes and performance records on animals. But there are some traits like feed efficiency that's rather difficult to collect the data. In disease resistance, well, animals have to be exposed to a pathogen to get the disease, and would prefer not to do that. So, but we can estimate the SNPs that are correlated to disease resistance and use those SNPs then to estimate genetic resistance for animals that have never been exposed to the disease. DNA tests can be done at a young age. In fact, they can take cells from embryos and do a DNA test on there. So, a lot of advantages here: enhanced accuracy, done at a young age, etc. Well, there are some disadvantages. It's a little bit costly, and on average, DNA test cost about $40 per animal. So generally, beef cattle and dairy cattle, an individual animal has more value, so it makes more sense to do DNA tests on cows rather than on pigs or sheep that have relatively less value on an individual basis. And to be accurate, you need thousands of DNA tested animals along with their phenotypic data to accurately know the value of these different SNPs. So it definitely, genomics gives an advantage to those breeds that have thousands and hundred thousands of animals compared to a relatively small breed. So it makes the popular breeds even more popular, I guess, because they can make more progress, and has definitely been somewhat at a disadvantage for the smaller breeds. Again, we can make a lot of progress if we just select generation after generation. And here again we've got two horses. These horses have the same ancestors. If you go a couple thousand years back in their pedigrees, in one case selecting for bigger horses, the other case smaller horses, and genetic selection is powerful, especially if you do select for good traits generation after generation. So that is the end of Genetics 8, Basis of Selection. You have a good day.